welcome everyone to our today's webinar. The topic of today is B-cell cell culture, and we want to give you advice how you can upgrade your B-cell culture. So let's directly start. So here I just have a short overview what we are going to talk about today. First, I'm going to give you an introduction on B-cells in general and why the B-cell culture is so important and maybe also so tricky. Then I will hand over to Henriette, who will give you some background information on the technology and why it is so important to have good tools for cell culture. And then in the end, I will give you some application examples on how you can standardize your B-cell expansion. So let's directly hop into the topic and start with the introduction on the B-cells. So as you probably all know, B-cells have very important functions in the immune system. So there are actually quite many. So they do produce antibodies and have a memory function. They produce cytokines. They play a role in wound healing, also in antigen presentation. And also an important topic is the Th1, Th2 cytokine balance. They also regulate dendritic cells. They play a role in tumor immunity. And then also, of course, co-stimulation is a very important topic. So there are actually, in general, two main subsets of B cells. So the B1 and the B2 B cells. So the B2 B cells are actually the B cells that we usually call B cells. It's the largest population of B cells, and it has a part in the adaptive immune system. So also we have here the B1 cells. The B1 cell is like more innate-like immune response. It is T cell independent. It has in general the same role as other B cells. They make antibodies. They also act as APCs, antigen presenting cells. But they really produce more, a natural type of antibodies. They participate in maintaining of the tissue homeostasis, as well as the immune defense against mucosal pathogens. But their repertoire of B-cell receptors or antibodies is more, polyspecific, polyclonal. So this way they also bind to self-antigens and can play a role in autoimmunity. And they also protect the body mainly against microbial antigens. And in these B1 cells, we have two main subsets, the B1A cells and the B1B cells. And now we look into the B2 cells. So from now on, I will just call them B cells and how they are developing. So mainly the B cells develop in the bone marrow. So from the progenitor cells, we have pre, immature B cells, then transitional B cells, and then in the end, the mature B cells. And in the bone marrow, there's a lot happening. So we have the VDJ rearrangement, so the rearrangement of all the genes until an actual functional B cell receptor is formed. There are also positive and negative selection is happening. What is really important, so the signals are mainly antigen independent, and also the negative selection is really important to have tolerance for the own body, of course. Then in the next step, the B cells, the mature B cells are mainly divided into follicular B cells and marginal zone B cells that can get activated. So here we have the follicular B cells that are getting activated. Those follicular B cells are mainly circulating in the blood. And after activation, they build a germinal center. And then affinity maturation is happening. So proliferation, maturation, apoptosis, and also here the stimulation of T cells is happening so that we always get the best highly affine, really good antibodies that we want to have. And out of here, we have the memory B cells that come out that can be activated upon antigen contact, that respond really fast and have a very high proliferation rate, that then are differentiating into plasma cells. So we have either long-lived plasma cells or plasmablasts that are then turning into other short-lived plasma cells that produce antibodies like crazy. But an activated B cell can also directly go into the status of the short-lived plasma cells to produce antibodies faster because, of course, this germinal center reaction takes a while. Also, we have here the marginal zone B cells. They really defend against blood-borne pathogens, and they have more T-cell independent plasma cell production, so they don't undergo this germinal center reaction. And then when they get activated, they also build plasmablasts that then turn into plasma cells. You see there's like a lot going on in the B cell development, and there are many different B cell subsets that we have. Also, we have the regulatory B cells. This is a very specific and very unusual subset of B cells, so it's still unknown where the origin actually is coming from. So some people think that it's like, specific pathway, and others think that with the right stimuli, actually each B cell can turn into a regulatory B cell. They have mainly immune suppressive functions, so they secrete cytokines like IL-10, and they could be generated in vitro through the addition of inflammatory signals, but there's still a lot of studying ongoing on this regulatory B cells. So here you can see why it is actually so hard to culture the B cells, because they really need several stimuli to really activate the B cells. And this is really hard to mimic in vitro, because in vivo there's a lot happening with the help of the T cells. So you have first the contact of the B cell with the B cell receptor to the antigen. And then in the next step, you also have the T cell that is then binding to the B cell. So we have the MHC presentation of the antigen of the B cell, then the T cell can bind with the T cell receptor to it, and then it presents the CD40 ligand to the B cells. And then, of course, also cytokine production of the T cells is happening. So they produce a lot of different cytokines that then bind to the cytokine receptor of the B cell surface, and what leads then to clonal expansion and differentiation, what really depends a bit on which cytokine mixture this T cell is actually giving and which signals this B cell is getting. So, and this to mimic this is really not so easy in culture conditions, because they like the friends of the T cells that giving them the help. So that was it so far from my side, from the introduction part. I will now hand over to Henriette so that she can explain you the technology background.
Thanks for handing over, Susanne. I will try to cover today a bit the technology part of the cell culture, and would like to have a look with you together on first factors influencing the reproducibility in cell culture, and then focus on one of those factors more in detail and try to show you how we can use this factor to increase the reproducibility in our cell culture. So let's have a look together. First of all, the cultivation can influence the reproducibility in your cell culture, meaning the media you are using, also the supplements like vitamins you are adding, also how you're subcultivating the cells, meaning are you trypsinating, in case of Ethereum cells, are you trypsinating the cells, how long trypsination goes, or are you using ecotase? And one of the biggest factors causing variability are animal-derived components like the FCS and variation between the different batches. Then we also have the cells per se influencing the reproducibility in our cell culture. In terms of cell lines, the age influences the outcome, as cell lines that are cultivated for longer time periods can display different behaviors or results as cell lines cultivated for a shorter time period. If we are working with primary cells, for sure the donor variability will influence our results, but also the way how we isolated the cells and how reproducible the procedure is will have an impact on our outcome. But also when we're thawing cells, this can influence the cell culture, meaning the time I need to thaw the cells, also the freezing medium I use, and how they are stored. If they're stored in liquid nitrogen at -80, this can also impact your results. And then the experimental setup will have an impact, meaning the timing of the experiment, and if the experimenter changes in between, this can also impact. But one factor which is also really important is the setup, meaning in terms of reagents you are adding and variance between the different batches. And this is one point I want to discuss with you today with more in detail. These are the cytokines we are adding to our cell culture for expansion, but also for the experiments, because cytokines can also have a great impact on your cell culture. And within the next slides, I would like to show you why this is, and how we can reduce this variability. So, if we are having 10 vials of a cytokine, different batches, and add of each vial 10 microgram to an assay, and for example, have a look at the proliferation of the cells, do you think it will have the same effect? Have all batches the same effect on our cells? If I ask like that, the answer is quite obvious. No, it's not. As you can see in this example down there. We will have cytokine batches with a low activity, causing less proliferation, and we have cytokine batches with a high activity, causing high proliferation. And this example, to my mind, shows nicely that weight is not equal to the activity for cytokines, meaning if I use the same amount of cytokine, it can have different activities. So why is this? Why is weight does not equal the activity for cytokines? For example, if we think about how proteins or recombinant proteins like cytokines are produced, here in the example of E. coli, they are denaturated. They need to be denaturated first, and then need to be refolded. But during the refolding process, we will also have misfolded or reaggregated proteins, which are not active anymore. So if we have a look now at our example for the cytokine with the low activity, most cytokines are misfolded and thus not functional. Whereas with the cytokine with a high activity, nearly all cytokines are correctly refolded and thus functional. And I think this example shows nicely why cytokine dosing should be done according to the activity of a cytokine and not using weight only. So to be able to dose to the activity, let's first think what biological activity for cytokine means. For cytokine, the ED50 value is defined as the concentration of a cytokine that is required to induce half of the maximal activity, and it's given in nanogram per milliliters. To make it more comprehensive, you can have a look at the example below. Here you can see a dose response curve where different concentrations of a cytokine were used, and at one point the response hits a plateau. That's the maximum response we get, and if we take 50% of this response, we will end up with the ED50 value. And this ED50 value is then taken to calculate the biological activity in units per milligram using the shown formula. How can you get this activity in your lab? Either you decide to do in-house testing, which means you will have to test every lot to lot by yourself, or you have to supplier doing the testing for you. But in this case, you need to be careful, because a lot of suppliers only state the minimal activity of the cytokines, meaning they guarantee you that the cytokine will have a certain activity, but don't tell you how active this lot is. For example, they guarantee you that the cytokine has an activity of 2 times 10 to the 6 units, but in fact, the batch you can get can have 2 times more, but also 10 times more. You will not know it. And with this, we can help you with our premium grade cytokines, because there we state you and give you in the COA the lot specific activity. And then you can use this lot specific activity to dose the cytokine in your experiments and do not need to do the lot testing anymore. So if we're using units to dose in our experiments, we should think about how reliable these values are. For example, if I use one cytokine and test it in two different assays, the results will not be the same, as the dose responses are influenced by the cell lines and the used assay conditions, meaning the units are only relative values. For example, if one supplier states he used one assay and the other supplier states he used another assay, those values are not comparable. How does it look like if the same assay is used? Yes, this makes it more comparable, but if I take one cytokine and test it in the same assay at different times or in different labs, the outcome can still be different, as the dose responses are influenced by cells, their cultivation and assay conditions. And that's the reason why assays should be calibrated whenever possible. Ideally, for this is a WHO standard taken, but such standards are not always available. If you use a standard for calibration, you also test it in the assay and then normalize the results of your assay to it. This will give you the calibrated activity and reduces the variability. If this is done, you can compare the results. And I want to show you within this slide how calibration reduces the variability. And the image on the lower left from in-house data from us, we can show how standardization can increase the lot-to-lot consistency. And then also from external results where different labs tested one cytokine and normalized it to an in-house standard. All labs used the same batch, whereas on the lower image, also the same batch of cytokine was used in all labs, but it was normalized to an international standard. And you can see how the reproducibility in the results is increased when an international standard, when the same standard is used in each lab. How can we find out if the assay was calibrated or not? This is usually stated on the webpage of the supplier or in the data sheet, and for us also in the certificate of analysis. There you can always see if the calibration to the standard NIBSC, if this is stated, the assay was calibrated. If we now have our calibrated units that we can use for our assay, we need to decide on the optimal amount that we are using in our assays. This is ideally tested one time in-house in your lab to define the optimal amount. To avoid all this lot-to-lot testing, it's also often done that just a high amount of cytokine is taken, which leads to an oversaturation of the experiment. But this oversaturation can be avoided if you know the units, the biological activity of your cytokine, and have tested once how many units you need to add to your experiment. This is shown on the example on the right. If you oversaturate, you can simply take 1000 units and you will end up in the plateau phase. But instead of 1000 units, you could also use 100 units and would get the same result, meaning you could save reagents and thus money if you dose according to the unit. That's the reason why it's important to know what you get. And besides the cost effect of oversaturation, it also has another dark side. Oversaturation can have a serious impact on the cells and the experimental results. As you can see here, with some publications given as an example, where it is shown that the concentration or the unit influence the results and can have a negative impact if you're not dosed correctly. To sum up, how we can support you with increasing the reproducibility in your cell culture are our premium grade cytokines. As we provide you with a lot-specific activity, you know what you get. Thereby, you can increase the reproducibility of your cell culture, save time, and reduce costs. And besides this, we offer a lot of our premium grade cytokines also as next GMP cytokines, which facilitates the seamless transition to the clinical research. And with this, I would like to hand over back to Susanne, that she explains you a bit more in detail the standardization of the B-cell expansion.
Perfect. Thank you very much, Henriette. So now we know all the details, what are the backgrounds, so what do we need to know to really do a proper and good cultivation. And now I'm going to give you the insights into the B cells. So there's a very standard protocol on how you can actually cultivate B cells. So you use the B cells, of course, mix it with CD40 ligand proteins and IL-4. And by this, you only get low expansion of B cells. But we actually made an optimized protocol. So this we have also available as a kit, so the B cell expansion kit for human B cells, where we actually have a mixture of different things that are important for a high expansion. So we have to optimize CD40 ligand multimeres across linking antibody and also IL-4 inside. And why is this so important? So actually, you see here on the picture that the CD40 ligand multimeres do not bind to only one CD40 receptor. No, it's binding to several ones. And that really makes sense, because when you imagine that usually the T cell would give this signal, would give this help to the B cell, it's not touching the B cell on just like, one side, on one receptor. It's touching them on several sides. And maybe also other T cells are coming and helping from the other side. So this really mimics the T cell help and leads to high expansion rate. This you can see here. So you really have like your B cells in the beginning, and then in the end you end up with like, highly expanded and activated B cells. So here I give you some details. So you will get up to 10-fold expansion of the B cells in vitro within 14 days. It is a standardized culture system with a very efficient B cell activation. So we also implement the max premium grade cytokines. So now you know why this is like so important, because Henriette explained that to you. And you also have an efficient activation of the B cells. So you really have an increased expression of the activation markers like CD86 and MHC2. Also, we preserve all the subsets with the kit, because it's really important, because you want to have all of them in the culture. So the subsets are distributed similar to actually the original fraction. And we also tested the B cell expansion kit in a modified version with IL-21 instead of IL-4, and we observed an earlier onset of activation and the differentiation into more mature and switch status. But I'm going to go into details right now so that you see really the data, what we actually measured. So here you see the expansion rates. So here we have the 10-fold expansion within 14 days with the normal B cell expansion kit, but it's already really nice. But when you add then the IL-21 to the culture instead of IL-4, they have to be clear, you see that the onset of expansion is a bit earlier. So you already get the 10-fold expansion after 10 days, and then after 14 days, both kits are more or less comparable. But we actually tested all of them. Here I show you the example for CD86 and MHC2 for both protocols. And you really see nicely here that with the standard B cell expansion kit, CD86 is highly expressed after 14 days of culture, and also MHC2. But with the modified B cell expansion with IL-21, you actually see already a high expression of MHC2 on the B cells already on day 7. What is like, going to, is a bit lower than after 14 days. But you really see that the B cells are nicely activated. And then, of course, I told you that all the subsets are, like, distribution is the same as the original fraction, so that we preserve the subsets. So here I just wanted to show you how we actually analyzed this. So we actually cultured the B cells for 14 days, as I described to you before, and did the flow analysis afterwards on the cells. And we first gated the dot plots on unstimulated B cells. So this is like really just the example how the unstimulated B cells look like. Excluded debris and doublets, and gated on viable CD19 cells. And we actually selected then here for the subset switched memory B cells, non-switched memory B cells, and naive B cells by analyzing for the markers IgD and CD27, so that you have the subsets here, switch memory, non-switch memory, and naive B cells. But we also checked for plasma cells and plasma blasts with the markers CD38 and CD138, what you can see here and here. So by this, we actually found out that the memory plasma cells have a similar distribution to the original fraction. So here you can see the B cell expansion kit, and next to it, unstimulated cells in the culture after 14 days. And here's the original fraction on day 0. And you see that actually the naive B cell population goes a bit down, but it is just due to the fact that they have a shorter survival rate in general. So they usually die after a while. And when you now have a look here on the right side, the modified B cell expansion protocol with IL-21, it looks a bit different. So you really have more switched memory B cells, and even plasma cells are popping up here. So you really see that we have a more mature, more switched status of the cells when you're using this protocol. And for this, you probably have to understand a bit what different cytokines are interested. So we also did cytokine secretion patterns. And I first wanted to explain you what actually these cytokines are going to do, so what we think they're doing. So we have IL-6, that's a pro-inflammatory cytokine that's actually secreted by activated B cells, so kind of a marker for activation. Then we have IL-10, we talked already about this in the very beginning, that is this is immune regulatory cytokine that is actually balancing the immune system, triggering antibody production, and really it's a counterbalance for inflammation. And we also have IL-4, what is the activation agent. So we told you already that usually Th2 cells to produce them to activate the B cells, but also this cytokine is activating the new naive CD4 T cells to get differentiated into Th2 responses. So in here we did the cytokine secretion patterns for our expanded B cells. So with the B cell expansion kit with using human IL-4, we have high levels, of course, of IL-4 and IL-6 on day 7. This level increased then over the time on day 14, what you see here. And then for this kit, the production of IL-10 is then increasing. But of course, here you have to be a bit careful to say that actually the B cells are producing the IL-4. Of course, they're not producing the IL-4 because it's rather the fact that we give it into the culture. Because when you now have a look on the results with the modified protocol with IL-21, you see that we have no IL-4 in the culture. So that really gives us a hint that the IL-4 for the other protocol is the IL-4 that we gave into the culture. But you really nicely also see that we have a lower secretion of IL-6, and that the secretion levels of IL-10 on day 7 are much higher than the other protocol. And this makes sense because it has been reported that actually they are in line with the plasma cell differentiation, because I already told you it's important for the counterbalance to the inflammation, triggers antibody production, and of course, which cells produce the antibodies? Yeah, it's the plasma cells. And then after 14 days with this protocol, actually we have low level of cytokines and more or less low level of IL-4. But of course, we have also other B cell stimuli, so we don't only have this one protocol, this one kit that you can use. We also have a lot of TLR ligands, FLT3 ligands. We have many cytokines. So Henriette already talked about that. So this is available for human and mouse. And also we have different sizes, different versions, so to say, of the CD40 ligand multimer kit that you can also nicely use for your culture protocol. And most of the cytokines and some also of the ligands are also available as GMP grade, what's really nice. So now I want to just give you a short side note on actually this TLR antagonists. So there are many different classes of the ODNs, and for B cells, mainly the class B ODNs and the class P ODNs are interesting. And there's like a slight difference in what they're actually doing to the B cells. So actually by this class B ODNs, you actually have a very low interferon alpha production, but a high IL-6 production, so high activation and proliferation of the B cells. And also we recognize that in recombinant cell lines, we have high NF-kappa B mediated signaling processes. With the class P ODNs, it's a bit different. So there we have high interferon alpha production and also high IL-6 production. And it's like mainly a more strong B cell activation. But for the NF-kappa B mediated signaling in recombinant cell lines, it looks the same as for the other TLR antagonists. So now we're already at the end. And I hope we could show you that we can really help you to improve your B cell culture. We offer a lot of different B cell stimuli. So if you're interested in just look on our webpage, there are a lot of different things that you can maybe use. We have the B cell expansion kit with a reproducible and really standardized culture process that preserves the subsets and gives efficient activation and proliferation of the B cells. And with the premium grade cytokines, you can never go wrong, because we have the lot-to-lot consistency, what reduces your cost, enhances the reproducibility, and allows standardization of your culture. Now we're already at the part where we thank you for your attention.